So that was a very enthusiastic presentation. Thank you. And now, so we are almost there. Now it's time for our actually last talk of the day with Professor Moin Saleem. He's professor of pediatric renal medicine and founder of PureSpring Therapeutics. He set up and had one of the world's largest renal disease research laboratories in Bristol, and he established the human conditionally immortalized glomerular cell lines as the gold standard tool for researchers worldwide. He founded a new gene therapy spin-out company with the largest ever Series A funding for UK University Biotech. Please welcome our keynote speaker, Professor Moin, and his presentation is titled Transferring Academic Innovation to a Successful Kidney Gene Therapy Biotech. Welcome. Here you have your
yeah, great stuff. Thank you very much for the kind introduction, and really nice to be here. So I'm an academic. I'm a pediatric nephrologist, so I treat children with kidney disease in Bristol, at Bristol Children's Hospital, and I also run a big glomerular disease research lab in Bristol. So I'm slightly unfamiliar with the surroundings of working with industry and pharma, and also venture capitalists. So I'm on a very steep learning curve. So I thought the best way of approaching this was actually to sort of give you an idea of the kind of journey that I've been through so far in getting this far and setting up PureSpring, and hopefully introduce you to a really exciting concept. So kidney disease, as I say, I'm a nephrologist. These are the patients that we deal with, and it's a huge global problem. And you may be aware that if you look at the incidence of chronic kidney disease worldwide, in populations, it's up to sort of 15 to 20% of the population, depending on how you define CKD. And it's a rising tide, so there are increasing numbers of deaths from kidney diseases. Some of the sort of estimated figures are given here. And also, it's an unmet need, but it's also a hugely expensive problem, because if you end up with end-stage kidney disease, you need dialysis, you need transplantation. Worldwide, that's not available to the majority of the world, in the developing world. So many patients die, and these kind of costs are mainly taken from the developed world. So the costs of end-stage renal disease globally and in the US is vast. But as I say, there's a big untapped and unmet need in the rest of the world as well. And the size of the market clearly depends where you start from, but it's, as I said, it's a rising tide, and there's increasing numbers, particularly in diseases like diabetic nephropathy. So the current epidemic of obesity, diabetes, etc., impacts on kidney disease as well. So the best way to understand it is firstly to understand the biology of the kidney, and I call the kidney the world's most sophisticated filter. It's not just a filter, it's responsible for homeostasis in lots and lots of different physiological body systems. And the nephron is the functioning unit of the kidney, as I'm sure you know, and we each have about 1 to 2 million nephrons in our normal kidneys. And at the head of the nephron is the glomerulus. So the glomerulus is a specialized capillary loop, so a highly specialized capillary loop, where arterial blood goes in and comes out, and filtration happens within that the capillaries in the loop. And so that's the glomerulus. That's the sort of functional unit of the kidney. And we know from figures from lots of sort of kidney registries across the world that 90% of end-stage renal disease originates in the glomerulus, and this gives you, and the central cell of the glomerulus, the cell the specialized cell, is called the podocyte, and I'll tell you a little bit more about that as we go along. If I stay in my comfort zone, I'll stay with the biology, but I will move out of that as well. So this is a kind of spectrum of diseases that affect the glomerulus, and it starts from early life with genetic conditions and several different or many different genetic conditions, to immunological based diseases. The kidney is a target of immune disease, hypertension, diabetic nephropathy. This isn't proportionate in terms of numbers, but diabetic nephropathy is by far the biggest group. And then aging, as you get older, you lose podocytes in particular, and you lose kidney function. So our challenge with PureSpring is to introduce new therapies to the kidney via the podocyte, which, as I say, is the central cell of the glomerulus, and using AAV technology, which is the approach we've used. And if successful, so the reason I've given that all that introduction is that the potential for curative therapy will then be there for a number of major kidney diseases, perhaps even the majority. So we spun out what we think is the world's first kidney gene therapy company, spun out of my university, University of Bristol, and a Series A investment from Syncona of 45 million. So we were very lucky to get a substantial investment. And I'm very British, and we don't like talking about money, particularly in academia as well. So I stole this slide from our CEO, Richard Francis. So he's more sort of bullish, and the vision is that if you can get, of course I'm bullish, but in a more British way. But the vision is that if this is successful, it can roll out really quite rapidly to indications and several different indications, which will be which will bring therapies to conditions, to a lot of conditions that simply don't have any therapies at the moment. We manage all these diseases symptomatically. There really isn't anything that's curative on the market. So let's get back to my comfort zone. Let's talk a bit more about the biology. So I've told you that it's the podocyte. What do we know about the podocyte? So we know it's the central cell of the filtration barrier. These are the capillary loops here in cartoon form. The capillary loop is made up of endothelial cells on the inside, a specialized basement membrane called the glomerular basement membrane, and then on the outside sits these cells called the podocytes. And this is a three-dimensional electron micrograph of podocytes. And you can see they're very beautiful cells. They look like octopuses, some people describe them. They have multiple branching foot processes and interdigitating what are called secondary foot processes. And if you do a cross-section across these foot processes, this is what you see. This is the blood side, this is the urine side, and blood filters through via endothelial cells, basement membrane, and then these are the interdigitating foot processes of the podocyte. And the dark areas here is the actin cytoskeleton. So the scaffold of the cell is made of actin, is made of an actin cytoskeleton, which is very highly and tightly regulated. And in between each foot process, so the cell-cell junction between the cells, is what's called the slit diaphragm. And here is where a huge amount of signaling goes on in a very sort of complex manner to regulate the dynamics of the actin cytoskeleton. So that's where the sort of control of the podocyte comes. And this is just a cartoon of the same slit diaphragm. So through studies of genetic disease, mainly familial disease, which causes podocyte damage and kidney disease, lots of proteins have been discovered, particularly that sit at the slit diaphragm, and some of them are completely podocyte-specific. So this one here, for example, nephrin, podocin, these are podocyte-specific proteins. If you get mutations in these, you get severe disease, which causes breakdown of the filtration barrier, and loss of proteinuria, which is the cardinal symptom of breakdown of the filtration barrier. And this sort of multi-protein complex signals to the slit diaphragm. It also does other has other key facets, such as attachment to the basement membrane and so on. So that's where what I call the sort of nerve center of the podocyte, where everything seems to be controlled. And this is just to illustrate, this is a review we wrote many years ago now, but to illustrate that the pathways downstream, we're understanding more and more from the slit diaphragm intracellularly, controlling, for example, there are small GTPases which control the actin pathway, but other key facets as well. One of the facets we've investigated over the years is that nephrin is key for insulin signaling within the podocyte, and clearly that's conceptually very important for diabetic nephropathy. So why Bristol Renal? Why did these people from Syncona come to us and say, we're interested in your technology? So over 20 years now, we've been pioneers and leaders in the field of podocyte biology. So that's been our main focus, and not just podocytes, but glomerular endothelial cells as well. So the main sort of cells of the filtration barrier. So we were the first to develop human podocyte cell lines, which are now the gold standard across academia, and all the major pharma across the world use our cells. We're one of the largest renal research groups in the world, which we've built up, centred on the glomerulus, with a reputation for both scientific innovation and, importantly, clinical and translational expertise as well. And so amongst the PIs in our group, at least half of them are also clinicians, and I think that's really important. And we continue innovation. We haven't just sort of sat on our laurels, so to speak, with the podocyte cell lines. We've made lots of relevant disease models, mainly with human cells. We've developed ex vivo permeability assays to test the function. We've developed a new 3D human glomerular spheroid, and we've also refined human organoids. So we're very good at the models systems, and that's really important when you're trying to develop these therapies. And we're also leading national and international cohorts of patients. Some of these diseases we treat are very rare, and it's really important to be able to be part of a network, a community, where you're interacting with these patients, you know where they are, and they're engaged with research. So just one more slide on why the podocyte is important. So hopefully by the end of this you'll believe me that the podocyte is important. So as I say, the vast majority of kidney disease originates in the glomerulus. And most glomerular diseases, and we've gradually over the years proven more and more with our models that we use, that the podocyte seems to be central for pretty much any disease of the glomerulus that you study. And some of the examples, I've mentioned diabetic nephropathy. We've worked very hard on proving that the podocyte is an insulin-sensitive cell, and crucially, when you damage those pathways, you get early diabetic nephropathy, so it's one of the initiating pathways of diabetic nephropathy. And then there are rarer diseases such as Fabry, FSGS, so focal sclerosis, which is a disease particularly sort of relevant to nephrologists because it's really difficult to treat. So lots and lots of diseases across the spectrum that are centered on the podocyte. And from the genetic perspective, there are now over 80 genes we know that affect the podocyte that can cause rare genetic forms of kidney disease. And that shows how complicated the network, how complicated the circuitry is in the podocyte that keeps it in its sort of functional specialized form. And these are just a few illustrations of the kind of things that the podocyte does biologically. So for example, it produces lots of complement factors, and this is a paper we published a couple of years ago. It secretes most of the important components of the basement membrane, the glomerular basement membrane, and there are genetic diseases like Alport disease that are disorders of that basement membrane. If you lose podocytes, you lose kidney function, and that very closely correlates with loss of kidney function. So loss of podocyte number in many, many conditions, not just Alport syndrome, but from aging to all kinds of diseases, correlates with loss of kidney function. And as I said, most of the genes that are implicated in this genetic forms of nephrotic syndrome, which is called steroid-resistant nephrotic syndrome, are expressed in the podocyte and highly expressed and functional in the podocyte. So let me just go through some of the data, just make sure I'm sticking to time. Some of the data that we developed that gave us our kind of pilot study and proof of principle. So this is treating this disorder called nephrotic syndrome. It's a disorder of the filtration barrier where you get heavy proteinuria because of damage to the filtration barrier and lots of different clinical morbidities. You may recognize Jonah Lomu, who's a very famous rugby player who unfortunately died from complications of nephrotic syndrome after a transplant. So this is the disease that we see in clinic. Some of these patients are sensitive to steroids, so it's an immune-mediated disease, but some of them are resistant, and this is the worst end of the spectrum. And in childhood, at least 30% of these patients have a genetic disorder. So for those patients, there's no effective therapies. They progress very rapidly to end-stage disease, and they need transplantation very early in life. And the most common gene affecting these children is NPHS2, which is a gene that encodes that protein I showed you on the cartoon, podocin, which sits at the slit diaphragm. So our hypothesis, and we started this project several years ago now with a PhD student, that AAV is an efficient, specific vector for gene therapy for these genetic forms of SRNS. And the premise is that the podocytes are terminally differentiated cells, so just like a neuron. So, and there are cell-specific promoters available. So if you can target the cell and you can get your gene into the cell, it should stay there for life and continue to work. So we worked on NPHS2, which encodes podocin. We used our human cell lines, so we had human cell lines, both wild type and cell lines we derived from patients with the commonest podocin mutation. And this is just, you know, summary of lots and lots of in vitro work, but we showed that we could transduce. We tried lots of different serotypes. We found one that transduced really well into podocytes. You can express podocin in the mutant cells, and you can rescue function in the cells in vitro. That these cells have a loss of adhesion, and if you transduce them with the wild type podocin gene, you can rescue or largely rescue that functional readout. So we needed to do the kind of definitive mouse experiment, and so we had an inducible podocin knockout mouse. So we induced disease by giving it tetracycline or doxycycline, and then we inject by tail vein the AAV vector, and we look at the phenotype over a number of weeks and take samples. And just one slide to sort of demonstrate the effect. So this is proteinuria. So as I said, the cardinal marker of kidney disease, of breakdown of filtration barriers, proteinuria. So that's an albumin-creatinine ratio. And as you can see in the saline-injected mice, they get massive proteinuria. In the mice that are injected with the wild-type podocin, and this is just trying two different promoters, human and mouse, and they had equal effects and significantly reduced proteinuria. And the mice that have saline die after about 100 days, and we got very good survival with the mouse that had been treated with the wild-type gene. So it shows in a very severe mouse model of disease, we can get really quite significant rescue. So that's the kind of principle on which we've built the company, if you like, that we can now target the podocyte, we can rescue a pretty severe disease in a mouse model. How do we sort of develop that now to take it to clinical use? So at the time when we had the data, we were approached by quite a few different companies who were interested, because nobody's ever done it in the kidney before. So it's a field that lots of companies were understandably interested in. And we talked to various pharma and rare disease companies, and they tend to be well-funded and different types of business models. And we've worked with industry over the years for lots of different projects, so they were pretty keen and they knew us already. But there were some drawbacks that we saw, some lack of experience, for example, different degrees of clarity on kind of scientific direction and ambition. And then there's the sort of group of venture capitalist investors who also have quite deep pockets, different models of working with academics. And again, I was very clear that I wanted to stay as part of this project as it moves on into the clinic. And we've got lots of the scientific experience, but also the clinical experience to be able to work with the patient groups and the clinicians. And these companies have varied levels of experience with gene therapy products. So Syncona were the ones that really sort of came out top, if you like. They've got a really good track record, and you guys probably know this better than I do. Early investment in gene therapy companies, taking them through to next stage capitalization. It's a small and focused team, very personable, good ability to work closely with the academics, which they've demonstrated throughout the stages of development. And the foresight to fund it all the way through, from proof of concept experiments to manufacturing to clinical trials. So we're not in the position where we have to sort of continue to raise money every 6 months or whatever. And they've got a very ambitious vision as well. So the vision that is developing still is applying the technology to a range of kidney diseases. We're not just starting with the rare or the ultra rare, but we want to include common alongside rarer indications, because we feel that if we can get the platform right, then we'd have the ability to go straight into commoner conditions as well. And then what about, what do they see in us that makes the partnership feasible? And I think it's very important that they saw we had a track record and leadership in the field, really solid proof of concept data, and really important to have representative disease models. And the disease models we've used are directly relevant to human disease. They replicate it almost exactly in the mouse models that we've used, and also the current human 3D models that we're using. The ability, we think, to drive the project, keep it going with all those models, and the kind of experience we've built up over the years with those. The clinical exposure, I think, is really important, and also patents being filed. So clearly you need a solid currency for commercialization, and we've filed several patents so far. So we've ended up with PureSpring. So Richard Francis is our CEO, a fantastic guy who came from Novartis and Biogen. That's me. We've got Julian and Ronnie, who are leading in process development and medical leads. And the kind of principle of the company is that we want to be the real innovators, we want to be the disruptors, and there's been a real lack of innovation in kidney disease in general. If you compare it to fields like cardiology and oncology, the number of clinical trials in kidney disease over the years has been really very poor. And the management of kidney disease is largely still with dialysis. As I say, there's no real curative therapies for the majority of kidney diseases. So we think what we're putting together is a platform across different to cover all the bases, so to speak. So the platform will be the same capsid, the same promoter, and the same route of administration, because we're just targeting one cell type, and we just need to get good at targeting that one cell. So we're engineering capsids. We've got proprietary promoters, and we're improving on those as well. We're working on a local delivery method in the mouse. I said we did tail vein, but that's because the mouse is so tiny we couldn't get into local delivery. But in large animals, we're working on local delivery methods, and we're working on in-house CMC to make sure that we get that right from the beginning. I won't say too much about the targeted delivery, but the principle is that you limit systemic exposure by going directly into the kidney. You need a lower dose, and you can optimize the efficacy. And we've already done experiments in large animals which shows this is feasible, and we're developing a kind of proprietary delivery methods for the kidney. And then just a quick word on fund cell. So this is a kind of parallel bit of work that gives us the ability to really sort of innovative discovery work in the kidney. So this was developed by Myra Ghiaccia at King's, who developed a kind of library of viral vectors that encode most of the secreted proteins in the genome. And then you can use that to test animal models of disease. He's done it in cardiovascular disease very successfully. And the idea is that this can be done in kidney models of, in animal models of kidney disease as well, which we have in-house. So you can screen for secreted factors. You use barcoding to actually discover which are the specific parts of the library that are causing the beneficial effect, they're causing the rescue. So you can screen for kidney diseases, look for rescue, and find out which particular viral vectors are causing that. So that gives us a number of programs in development. At the moment, we've got 3 main programs. We've got the fund cell program, and we're still at relatively early stages. So, discovery to lead candidate. Hopefully within the next 3 to 6 months, we should have lead candidates in at least 2 or if not 3 of these programs. And just a sort of quick overview towards the end of the kidney therapeutic landscape. So at the moment, as I say, there's not much out there in terms of real innovative ways of targeting kidney disease, and there's a few small molecules in trials, some biologics, but we are the only people in the gene therapy space at the moment, and I think it should be a very sort of valuable field to move forward. And again, you guys will know this much better than me, but AAV gene therapy landscape clearly has not is very active at the moment, but has not addressed kidney disease so far. So we're hoping to be part of this kind of slide in the very near future. So that's just a sort of summary of what I've just said, and I'll just sort of wrap up. Oops, I should have a thank you slide, but seems to have disappeared. I'll thank everybody. There's a lot of people, and thank you for your attention.